At this point, I'm happy to introduce kind of a novel dimension to this symposium. As co-leader of the Education and Training Work Group, I've worked to really ensure that we have a solid training experience for early career investigators. As you know, we funded 12 Scholars Award to early career researchers this past year. In addition to having them take advantage of some of the educational opportunities within the Michigan Institute for Clinical and Health Research, as well as serving as guest lecturers for our new Precision Health undergraduate elective, we're having them today compete and talk about their science in a very succinct manner. Additionally, you'll find posters if you want a little bit more information during our poster session, and we really encourage you to seek them out and ask them questions. So the competition program that we're debuting today is designed to really enhance research communication. It's based on the 3-minute Thesis program developed by the University of Queensland. The competition will engage 8 of our funded scholars. Each will have 3 minutes to discuss their science in 1 slide. For those of you that are not presenting, take a deep breath and say thank you. For those of you that are presenting, just take a deep breath. So they're going to provide a very concise and intriguing, engaging description of their funded research project. They are going to be judged. We have 3 guest judges today. Dr. Gretchen Piet, who is director of our new graduate certificate program, Dr. David Cantor, who is a professor emeritus here at the University of Michigan, and Dr. Eric Mikkelsen, who is one of the co-directors of the Precision Health Initiative. And additionally, we are going to have a People's Choice Award. So you may have noticed these table tents. Please read them because they do have your instructions on how you can vote not only for our People's Choice Awards, but also for our poster awards. You have ballots on your table, and there should be ballots equal to the number of seats at your table. Please don't stuff the ballot box. Please just use an honor system and only include 1 ballot. The speaker ballots are on white paper. The poster ballots are on different colored paper. The prizes are substantial. The first prize winner is going to receive $1,000 in research travel funds. And for many who are graduate students, this is a substantial award. We are going to be hearing 8 scholars throughout the day in groupings of 2 or 3 at a time. We were going to be beginning today with Nina Aguchukwu, but she had a baby 2 days ago, so she sends her apologies in not being able to present. We actually had to tell her to stay home. So to start off our 3-minute thesis competition, we have Neriman Tokan.
I think we're fine. Hello. I'm Neriman Tokcan. I'm a postdoctoral researcher in the Department of Mathematics. So let's get started. With the recent developments in information theory and technology, we are now able to collect and store data from multiple sources at the same time. We now have access to very complex multidimensional data, and it brings some challenges. So I work on an interdisciplinary research project on classification of cardiac index from patient data with multiple modes. And when we analyze this data, we have lots of challenges on the way. First challenge is how to represent this data. Because like most of the time, we represent data with vectors or matrices, but this has multi-modes, and it's a multidimensional data. That's why we now represent them with tensors, which is generalization of matrices. Now you can see these lovely cubes. We are representing them with cubes. And OK, we are done with the first challenge. What about the second one? How to analyze this data? Lots of data analysis methods, they are based on matrix representation. Now, our theoretical challenge is how to generalize matrix-based methods to higher dimension. For example, machine learning methods, data analysis methods, they utilize matrix factorization. Now, we are trying to generalize matrix factorization to tensor decomposition. What is the common approach for tensor decomposition? People take the tensor. They don't know what it is. They say, I'm going to slice you. I'm going to get matrices because I know what's a matrix, and they continue from there. But if you do like that, you are going to lose all the information between different slices. So our idea is, don't slice your data. Don't be a ninja, right? Then what we are trying to do, we develop a tensor decomposition method. We decompose our data to meaningful parts, but we keep the integrity of the structure. And second thing, there are similarity scores to compare matrices. We are finding a generalization. Now we are comparing multidimensional data. We develop a tensor similarity score. This research still requires a lot of theoretical effort and collaboration between different disciplines. And as a mathematician, my main goal is to build a bridge between theoretical side and application side. And I believe that if researchers from different areas, they get together, and if they share their ideas, they can target more challenging problems. And in this case, we can make fascinating discoveries. Thank you.
So our next scholar is Abhinav Unchiriju.
Cancer is a dangerous disease that has touched millions of lives and is getting more and more difficult to treat. But what if we could learn from cancer patients of the past to discover new and more efficacious treatments for cancer patients in the future? Good morning, I'm Abhinav. I'm a postdoc in the biomedical engineering department, and I'm trying to answer that very question. Decades of research tells us that several factors can lead to alterations in the DNA of healthy cells. A very small fraction of these alterations can lead to transformative characteristics or hallmarks that turn a healthy cell into a cancerous one. Some of these we know are pretty common. Cancer cells can grow uncontrollably, and they can spread across the body. But one such hallmark is of particular interest to us: altered cellular metabolism. Because metabolism is the engine that drives not only essential cellular function, but also several pathogenic traits in cancer. My overarching aim is to discover or identify these metabolic traits specific to cancer and exploit them for therapeutic benefit. Although cancer research has come a long way, treatment is difficult for one significant reason, and that is tumor heterogeneity. And this heterogeneity not only exists across patients with the same cancer, but also within the same patient as the disease progresses and the tumor acquires more mutations. Current chemotherapies paint in broad strokes. They tend to lump patients with similar parameters and treat them similarly. And unfortunately, they're not 100% efficacious, and they are accompanied with these debilitating side effects because of their effect on healthy cells. Our solution involves using the swaths of data oncologists have collected from cancer patients. And in particular, we care about mutational signatures from these cancer patients that can capture the heterogeneity I spoke about. I focus on a particular type of mutation that deals with deletions in regions of the chromosome. These deletions in the genetic code are beneficial to cancer cells because they get rid of genes that can actually suppress cancer growth. But there's always collateral damage. And sometimes they lose genes that are required for essential cellular metabolic function. But human biology is resilient. It can activate these redundancies that can help cancer cells survive. But that's like looking for a needle in a haystack. So we built a computational algorithm to look for that needle. Our hypothesis is that if we target those metabolic redundancies in cancers with particular mutation signatures, we can target cancer cells more effectively while minimally affecting healthy cells. Our hope is that a paradigm such as this could transform cancer treatment, make it more precise, and improve the quality of life for cancer patients. Thank you.
We are going to continue now with our next three scholars, and the person that's going to be kicking off the event is John Plass.
Okay, hi, I'm John Plass from the Department of Psychology. Primary brain tumors have low survival rates and can interfere with brain function, reducing patient quality of life. For example, patients with glioblastomas typically survive for 1 year or less and may develop seizures or neural deficits due to brain damage and swelling. Tumor resection can mitigate these issues and prolong life, but must be performed as aggressively as possible to prevent tumor regrowth. To maximize the extent of resection while retaining brain functions, intraoperative brain mapping is used to identify function-critical areas for preservation during surgery. The current gold standard for pre-surgical brain mapping involves disrupting brain activity with electrical stimulation while patients perform diagnostic tasks like picture naming or word repetition. If performance declines during stimulation of a certain site, the site is deemed to be task critical and preserved during surgery. One weakness of this approach is that it requires sufficient baseline performance for comparison, which may be impossible when tumor pathology interferes with task performance. Moreover, stimulation does not discriminate between different subfunctions that could be disrupted to produce task deficits, but different subfunctions may be prioritized differently for resection or preservation. To address these weaknesses, we propose to use intracranial recordings of task-related brain activity as an alternative approach for pre-surgical brain mapping. We hypothesize that intracranial ECOG recordings might facilitate brain mapping by providing detailed electrophysiological information about brain function without relying on patient performance metrics. To test the feasibility of this approach, we recruited patients undergoing tumor surgery at Michigan Medicine and UC San Francisco. And to assess whether ECOG could be used to discriminate between different subfunctions involved in speech production, we tested whether the timing of two signals, the high gamma response on the top left here, and beta suppression, could be used to discriminate between areas involved in premotor planning and primary motor execution. This is important because resecting primary motor areas but not premotor areas can produce long-term speech deficits. As shown on the left, the timing of the high gamma response, an index of neural firing, was very effective at discriminating between premotor areas, which showed maximal activity before speech onset, and motor execution areas, which showed maximal activity during speech. That's in the orange and red, and the premotor activity is in blue. Next, to assess whether ECOG-based mapping agreed with stimulation-based mapping, we compared ECOG high gamma responses to stimulation mapping results. Two example patients are shown on the right here. We found that electrodes exhibiting the strongest high gamma response, which is marked with the solid circles here, tended to cluster near task-critical sites that were identified with stimulation mapping, which is marked by the yellow stars here, which suggests that ECOG could accurately localize task-critical areas. And moreover, the high gamma response allowed for premotor and primary motor sites to be discriminated, and for additional sites exhibiting weaker activity to be identified. That's the semi-transparent circles here. Thus, our preliminary results suggest that ECOG-based mapping may provide a valuable alternative or supplement to stimulation-based mapping, and we're currently validating this approach with a wider variety of tasking patients, and we aim to make these tools available to clinicians upon further validations. Thank you.
And then continuing on with our 3-minute thesis, we have Aaron Morris.
Hi, I'm Aaron Morris from the Shea Lab in Biomedical Engineering. So the focus of my work is autoimmune diseases. So autoimmune diseases are when your body's defense, the part of your body that's designed to fight pathogens and kill off invaders, goes awry and begins to attack your own tissues. And this can really happen to any tissue in your body. I have some examples up there. I focus mostly on type 1 diabetes, so that's going to be an autoimmune disease of the pancreas, as well as multiple sclerosis, so that's the brain and the spinal cord. And so one big problem with autoimmune diseases is, although genomics and precision medicine have helped us identify patients that are at higher risk for autoimmune diseases, we often don't know when they're going to start or if they're going to start. And so what we would love to do is to biopsy the tissues that we're interested in to see if an autoimmune disease is about to happen there. But we can't biopsy patients' brain and spinal cord all the time. So how are we going to do this? Well, some people have tried to make blood panels. So they take blood from a patient and try to identify whether the patient's going to have an autoimmune disease or not. But these haven't translated very well to the clinic. And one of the reasons for this is likely due to the fact that the population of cells that causes the disease in the blood is quite rare. The other thing is when the cells are in the blood, they don't have the same phenotype that they do when they're in an inflamed tissue. And so what we wanted to do, and what the focus of my work is to try to use tissue engineering approaches to create an artificial site for biopsy. So basically, can we take these porous implants that you can see in A and B there, and implant them under the skin in a mouse to allow them to form an inflamed tissue so that we can biopsy them and get information about the disease over time. And so we implant these scaffolds, we induce the disease, and then we can take biopsies of these scaffolds and analyze the transcriptome at them. And then we can computationally process this data and come up with scores of disease. And basically what you see in the bottom graph here is the black points are healthy mice, so they all have relatively low scores. And then the other colors are diseased mice, and so their scores are much higher. And importantly, the yellow is actually before the mice show any outward symptoms of disease. So we're able to predict the onset of disease, in this case, a mouse model of multiple sclerosis. We can go beyond this, though, and we can actually, in a relapsing remitting model, predict the relapses before they occur. Excuse me. And we can predict the response to treatment. So we're able to identify treatments that will work or not work in a particular mouse. And then we've extended this beyond multiple sclerosis. So this also works in type 1 diabetes, and we are kind of ramping this up for other efforts. So thank you.
And then last for this session, we have Isaac Zia.
So this is Dr. Richard Boland. He used to be a faculty member at U of M. When he started medical school, he was convinced that he's not going to live very long because you see both his father and grandfather died of colon cancer in their 40s. He didn't know what's wrong with his family until many years later, through the work of Dr. Henry Lynch and others. His family has a pathogenic MHC2 gene. As of today, Lynch syndrome is the most prevalent cancer syndrome in the general population. 1 in 300 people have Lynch syndrome mutations. The problem is, it's not always very straightforward to tell whether a mutation is benign or pathogenic. In fact, 93% of the mutation discovered in patients right now are classified as variants of uncertain significance. When patients are told that their case is uncertain, that's not precision health. In my mind, in the future, for precision diagnosis, when patients present a mutation, we should be able to tell them whether they have Lynch syndrome or not. So my research project, funded by Precision Health Scholarship, aim to address this uncertainty. So using state-of-the-art mutagenesis and massively parallel sequencing technology, we produced a map showing pathogenetic score for every single possible, mathematically possible mutation on MHC2. So MHC2 encodes 934 amino acid. On x-axis here, the native residue and their positions are shown. On y-axis, these are the 20 common amino acids. Each spot here represents that specific mutation. So in the future, when a patient's genetic testing is done, doctor can just look up this table and see if the mutation corresponds to a blue spot here, patients can pretty much be sent home worry-free. If otherwise it's red spots, a management plan can be developed to monitor the health of the patients. So we developed this assay specifically for Lynch syndrome, but our research approach can also be applied to other human disease-related genes. Thank you.
So the final round, our first speaker is Arun Submanayanan.
Thank you, Vicky. Hello, everyone. My name is Arun. I'm a PhD student in the Department of Computer Science and Engineering, advised by Professor Rituparna Das. Today, I'll be speaking about the computational needs of whole genome sequencing and why the general purpose CPUs that we all have and love may not be able to keep pace with sequencing data. Today, less than 0.1% of the world's population have had their genome sequenced, but this is expected to change soon. The cost of sequencing a human genome has dropped dramatically from about $1 million in 2008 to less than a few $1,000s today. And it's expected that Illumina will produce a $100 genome soon. So that means that genome testing could become as affordable as a routine blood test. This dramatic reduction in cost has somehow contributed to a DNA data deluge that we are unable to keep up with. Genomic data is expected to grow at the rate of 2x every 6 months. Historically, our computational hardware has been following Moore's law and doubling in performance once every 18 months. But unfortunately, even Moore's law is coming to an end because transistors can no longer be made any more smaller. And that means now we are heading towards a trend in which performance will double only once every 20 years. So the end of Moore's law is worrying because now we have a DNA data deluge, as I said before, and we are not able to keep up with. So consider a typical sequence analysis pipeline. A sequencing machine produces around 350 genomes every week. And a high-performance CPU is only able to analyze 17 genomes in a week. And this is going to cost you around $50 for a single genome and take more than 10 hours. The goal of my research is to bridge this bottleneck between sequencing and analysis by using specialized hardware. So what is specialized hardware? Specialized hardware is hardware that is customized for a particular task, and it can deliver orders of magnitude better performance and energy efficiency at a fraction of the cost. We are working towards developing an FPGA-based genomic data analysis service in the cloud that we believe could reduce the cost for analyzing a human genome from about $40 to less than $1 and reduce the time from 10 hours or so to less than 20 minutes. We have explored the computational aspects of the secondary analysis pipeline. We are very excited about specialized hardware, and I'm hoping to learn more from you about the bottlenecks in your own pipelines. With that, I'll end. Thank you.
Thank you, Arun. And the next scholar that is going to be speaking to us today is Andy Canny.
Thank you, Vicky. Thanks, everybody. I'm Andy Canny in the pathology department here at the University of Michigan. So we work on prostate cancer. Prostate cancer, some of you may know the statistics, #2 killer, #1 most common cancer. It's very common. Some of the low-grade disease can be relatively indolent, so it relies on use of good biomarkers in order to detect or be able to tell the difference between aggressive disease versus indolent disease. One of the biomarkers that's commonly used, and some may have already had this done, is serum PSA on the right side here. So it's a relatively okay biomarker, but it has low specificity because there are other factors that can increase serum PSA, such as inflammation of the prostate and so on. So what we set out to do is use the genomic landscape and that set of genomic and transcriptomic aberrations that drive prostate cancer in order to have better biomarkers, to have a test that uses a multiplex set of biomarkers that can refine that risk prediction and improve compared to PSA. And we're uniquely positioned to do that because much of the genomic landscape of prostate cancer was discovered in our group and here at Michigan. Prominent among those are ETS gene fusions, which are shown here. You can see the mouse. ETS gene fusions, which fuse a gene that's under the control of the androgen receptor, which is very important in prostate cancer, to an oncogenic transcription factor. There are fusion-negative cancers, which have different mutations, copy number alterations, and so on. So we wanted to put together an assay that combines all of those in order to detect prostate cancer in urine after a digital rectal exam, which the doctor is going to perform anyway, but this is supposed to enrich for prostate contents in the urine. So we put together a panel of next-generation sequencing RNA panel that targets a number of these. We trained a model and validated it in a couple of different validation cohorts where our area under the ROC curve performed or was higher than PSA alone. So this looks very promising. We are also are working on a version of this that removes the need for a digital rectal exam. And the data for that are somewhat encouraging right now. High-level transcripts are detected pretty well, but low-level ones may need some optimization. So in conclusion, we're pretty excited about this. We've disclosed it to the university for intellectual property, and we hope to see it all the way to the clinic, where one day this assay can give a urologist or a family doctor a better tool, a non-invasive way to tell whether to send the patient for a biopsy or not. Thank you.
And then our last scholar's presentation is Ida Sureka.
So the last but hopefully not the least. So I'm Ida Surakka. I'm a postdoc in Kristen Willer's lab in the Department of Internal Medicine. Background is in biostats, and I'm working on the genetics of cardiovascular diseases. So cardiovascular diseases are the #1 cause of death worldwide. It's also a complex disease, which means that both genetic and environmental factors are underlying the disease risk. In the current clinical practice, the high-risk individuals are usually identified through different types of laboratory measurements, like high cholesterol levels or high blood pressure, also through things like obesity and unhealthy lifestyle, which means that they are usually identified in their mid-age in the annual health checkups, when there's also a chance for irreversible damage already being done to the patient's arteries. Genetic information, which accounts for a bit over half of the risk of cardiovascular diseases, is not used in the clinical practice as of now, which is kind of bad, because genetic risk factors, they are the same since the birth to the death of the individual, which also allows for the earlier detection of the high-risk individuals. Also, inclusion of the genetic risk factors of the lifestyle factors, like, let's say, cholesterol levels or blood pressure to the models, could increase our prediction of the heart disease risks. And also, we could use those like lifestyle risk factor, genetic risk factors in the dissection of the CAD risk, which could be then used as an in the prevention of the disease. So my project is aiming to build a web application using all of these 3 different groups of risk factors, so CAD, genetic risk factors, lifestyle factor, genetics, and also the laboratory measurements, and to create a web application that could be potentially used in clinical practice in order to identify the high-risk individuals earlier. Thank you.
Thank you very much. So now is the time for you to fill out your white ballot, and we will have people going around who will collect the ballot. Check one box only in terms of the People's Choice Award for the scholar presentations. Now we are on to our awards presentation, and we have 9 awards, in fact, that we're going to be giving. So I will talk about the categories, and then I will name the first and second place winners. So if you hear your name called, please come to the stage to get your award and pose for a photo. I'm wondering if I could ask our 2 co-directors of Precision Health if they would be willing to be in here as part of the photos with our scholars. So our first award today is for our poster session and is for our most interdisciplinary poster. And our second place award winner goes to Stephanie Belis and Stephen Parker. Congratulations. And our first place award goes to Scott Peltier and Ben Hampstead. Congratulations. Our next award category is Most Potential to be Transformative. And our second place winner is Scott Peltier and Ben Hampstead. Congratulations. And our first place winner is Sirim Chandrakarna, who was unable to be here today, and so A. Sam Berry is the one that presented. Congratulations. Our next award is the Scholars Poster Ballot, the Most Potential to be Transformative, and our second place is Aaron Morris. Congratulations. And our first place is Abhinachav Atrija. And then the last three categories that we have are our 3-minute thesis competition. Our second place winner in the 3-minute thesis is Abhinachav Atrija. He has also won the People's Choice Award as well. And the first place winner of our 3-minute thesis competition is Aaron Morris. And with that, we are concluded. Please join us for the networking reception, and I look forward to seeing you next year at our Precision Health Symposium.